Hi, it's Peter Baggs. This is lecture two. It's actually dealing with material that is largely, many of the figures that I'm going to be presenting are actually from Stein's book, chapter one. Before we start, I just want to very quickly go over a couple of admin issues. First is we did a poll, and I thank all the students for replying to the request. About 30 to 35 students actually replied. And the question was, how should we proceed with lectures? And the decision is that we will have asynchronous lectures provided hopefully 48 hours before the planned lecture period, which is on Tuesdays and Thursdays. Those periods, the Tuesday, Thursday, 1 to 2:30, will be the synchronous lecture, referred to as the synchronous lectures, and they will be designed, and the focus will be on discussing material that's in the asynchronous lectures as well as in the class notes. So it won't be a formal lecture period. The other thing that came up was some questions related to what was written in the syllabus, incorrectly written in the syllabus, and that had to do with if you miss a midterm. So if you miss a midterm, you obviously, you know the deal. You have to get the proper documentation. Provided you do that, then if you miss the midterm, there will not be a makeup midterm. Your final exam will then be worth 40%. So the portion of the midterm will be actually transferred to the final. Your final will then be worth 40%. Something that did not come up in the synchronous lecture yesterday is is what I need to emphasize now, and that is you're responsible for what's in the notes. That's going to be most of what you really need to focus on. But there's going to be things that come up during the lecture period. So I'm going to introduce things occasionally in the lecture period and go a little bit beyond what's in the notes. You're responsible for what's in lecture and in the lecture notes. Problem sets are designed to really reinforce ideas in the lectures and in the lecture notes. So there won't really be any questions directly related to that, but certainly a good understanding and knowledge of the problem set material would be very helpful for you as you go through forward in this course. Okay, so what we're going to cover today is actually primarily the structure of membranes. That's really where we're trying to go. I'm going to give you a little bit of the history of the discovery of membranes, but most of it's going to be talking about the biochemistry, the biochemical makeup, and the structure of the membranes, and we're going to end up talking about the mosaic model, which means it's a lipid bilayer with implanted membrane proteins. That's what mosaic means. It's got two elements to it, and those are the two elements: a hydrophobic lipid bilayer and membrane proteins. And we'll talk a little bit along the way about functional consequences, because in fact, many of the seminal discoveries related to membranes that led to, the establishment of membranes as being essential for cells and life really came from functional studies. So we will touch on those very, very superficially as we actually move along here. So I want to start with this because it's sort of current, right? We know what membranes do, right? They separate inside from outside, and we typically associate that with cells, right? Like a cell has an inside and an outside, and what separates inside from outside is actually the cell membrane. But virtually every form of life, except a few, actually have membranes, and that includes enveloped viruses. And I think those are very relevant right now because the examples I'm giving you here are COVID-19 and the influenza virus. Both of these are what are referred to as enveloped viruses, which means they have membranes. And you can see from these illustrative diagrammatic representations of these viruses that they have this coat around them, this planar circular coat, which is the membrane. That's this right here and right here. And then projecting from that are these big red things. And we know that that's actually the spike protein in the COVID, if you've been following that story at all. And whereas in the influenza viruses, they actually have a diverse group of proteins that actually are involved in their infectivity. In both cases, the membrane actually, encapsulates the genetic information, which is actually RNA in both cases. This is a single strand RNA, and I believe influenzas have 7 chromosomes comprised of RNA. And along with a number of enzymes, right, that actually are necessary for the early replication and the early usurping of control of cells in which they actually infect. Um, and an important feature of these types of viruses is illustrated by the fact that both of these kinds of viruses, you can reduce your susceptibility to them very, very significantly by the frequent use of organic solvents, like washing your hands, right? You're constantly told that using the solvents, these solvents that are provided now, every time you walk into a building, these are actually soaps and very often organic solvents. And both of those actually turn out to dissolve membranes, right? And so you're basically destroying the ability of the virus to control its internal environment. And that's how we actually protect ourselves from these particular viruses. So that's just, an illustration of the importance of them. Now, when we think about membranes, they're synonymous with cells. And cells were actually, discovered a long time ago, and I'll talk about that in a moment. But we know that the cell is sort of today from your cell biology, that it forms the outer perimeter of the cell, and that's why we call it the cell membrane. Or it's also called the plasmalemma, or in the case of muscle cells, it's called the sarcolemma. And that refers actually to the outer membrane, right? So that's what's separating, the outer cell from the inner cell, self from non-self. But, if you look inside cells, there's actually a whole host of different kinds of membrane sacs within cells. For example, the sarcoplasmic reticulum, which is very similar to the endoplasmic reticulum and the Golgi. In fact, they're physically connected to each other in most cells. There's the nuclear membrane, there's a mitochondrial membrane, there's lysosomal membranes, there's synaptic membranes, there's endocytic vesicular membranes. And these obviously are not separating self from non-self in the cell, but they're actually encapsulating or enclosing very, very unique, distinct environments separate from the cytoplasm. For example, the SR has in muscle cells is filled with calcium. It has about 10 millimolar of calcium, although most of that is actually bound, maybe about a half a millimolar free calcium, et cetera, right? These all have very, very distinct intra-membranous environments that actually separate inside of the sac from the cytoplasm in that case. Now, the idea of the concept of cells actually comes from experiments done way back in 1665 by Hoek, and he was using Van Leeuwenhoek's microscopes. And what he saw and what he looked at initially in his first description of what led to his cell theory was actually a cork tree. And he did cross sections of these and was able with the microscope to see that there's these, very clear sort of honeycomb structures to the cork tree. And he referred to those individual units as cells. He actually thought they were empty. And there was a theory at that time that plants were this fibrous material and inside this fiber was actually just filled with water. Turned out that obviously wasn't correct, but that was the initial thinking, and that's where the cell theory was first articulated. Van Leeuwenhoek actually also saw this in ash trees as well as a whole bunch of other stuff. He published hundreds, maybe even thousands, I don't know how many, I can't remember exactly, but it's certainly hundreds of papers because he looked at everything. He looked at people's shoes, at, trees, leaves, he looked at everything and put it under the microscope. And he's the first person to describe many of the microbes that we know about today, as well as fungi and a number of other things. And collectively, they gave this gave rise to this idea, right, that there's an underlying pattern and structure to many of the life systems that actually that one could actually look at. Um, yeah, so that was 300 years ago, and it wasn't until much, much later that we actually realized, or that we've become aware of the fact that what actually is the distinguishing feature of the boundary of the cell is actually not the cell wall, which is actually, I forgot to mention, which is what Hoek was and Van Leeuwenhoek were actually looking at. It was much, much later that it was recognized that there must be something like a plasma membrane, an actual membrane, a fluid mosaic model membrane. And it really wasn't until 1972, right, which is very, very recent relative to the work of Hoek and Van Leeuwenhoek, before we actually recognized what we actually now know. Yeah, and it wasn't until the 1830s that Schleiden and Schwann actually proposed that the cell theory was also applicable to animals and members of the animal kingdom. And much of their work was actually based on developmental biology. And essentially the thinking was, and what they were looking at was, the development of a frog from a single egg to an entire organism. And so when they put things under the microscope, they realized that these different tissues had clear separations between, units that looked like cells, like what we're seeing in plants. But even then, the concept of a membrane separating these different entities, these cellular entities, was still not even conceived of, frankly, at the time. And it really wasn't until much later that the idea of membranes actually became apparent. I'm going to show you a couple of figures from one figure actually from Lombard's paper in Biology Direct in 2014, where he gives a history of the membrane. You might want to consult that. It's kind of an interesting read. And what he points out is the fact that, for years after Schwann had suggested that animals were also comprised of cells, the idea of a membrane, as I've already said, was not apparent. And the sort of theory that was accepted for a very long period of time was this idea that a cell was actually comprised of something called a protoplasm. And a protoplasm is a kind of like a form of matter that is sort of self-contained. And the idea was based on, phases of matter. We know that there's ice and there's fluids and there's gases, and you have these different phases. And so it was believed at one time that these were special phases of matter that were sort of self-contained. And it wasn't until much later that the existence of membranes actually was proposed and established. And much of that actually starts with the functional studies of Van Hoff and Nernst in the late 1800s. And the area I'm talking about is right here from Lombard's paper. And their work led to the, theories and ideas, and they were looking actually at living organisms, both of them, when they came up with their ideas on osmosis in the case of Van Hoff, and of electrochemistry was Nernst's work. And they didn't really put it together. The person who actually put it together was a person by the name of Bernstein, and he used Van Hoff's and Nernst's ideas to actually perform a series of experiments, functional experiments, that could really only be explained if you thought about there being essentially a separation between the inside and the outside of a cell. And he's the first person that proposed this idea that there must be a physical barrier, a semi-permeable barrier. And the properties of that barrier, his work clearly showed, was that it was permeable to hydrophobic substances and relatively impermeable to ionic substances. And that's Bernstein right here at this particular time frame, just at the turn of the 20th century, the beginning of the 1900s. And at the same time, working in a very parallel way, was Overton. And Overton was actually more of a biochemist. And he was interested in membranes, and he was doing things like dissolving them and looking at these properties. And he recognized and concluded that that these membranes were actually phospholipids. And he even suggested that the phospholipids actually form a lipid bilayer. This again at the turn of the century. So essentially, what Bernstein's ideas led to a person by the name of Huber right around the same time. And he suggested, taking into account Bernstein's functional measurements, that the membrane must be a mosaic. And the idea is actually, the diagram of his is actually shown here. And it clearly shows this idea that you have to have some protein and you have to have some lipid. And this is Overton's lipid bilayer. The idea was that essentially that these membranes he knew were permeable to hydrophobic substances, and so they had to have some kind of lipid properties. But they formed a physical barrier, right? And then on top of that, there was relatively low permeability of these barriers to sodium, but at the same time, there is a high permeability to potassium. So Huber actually suggested that these things might actually be proteins that are also working along with the lipid. And that's where the idea of a mosaic actually came from. And this was actually his rendition of that. He thought maybe the protein was somehow on the outside and somehow able to move, ions across this physical barrier that actually developed. There's a set of experiments done in the 1920s by a man by the name of Frick, who, now my pen's not writing, person by the name of Frick, who actually performed capacitance measurements. And those were very definitive because he was able to show that he could very rapidly change the amount of charge inside a cell and hold it there for long periods of time. And that really could only happen if there was some kind of physical barrier for diffusion that actually existed. So his work was kind of like the final nail in the coffin to show, yes, there's a physical barrier between the inside and the outside, and it has these properties suggestive of a mosaic that actually must exist within the system. But the real definitive studies establishing the presence of the membrane in cells had to wait until the invention of the electron microscope. There are lots of indirect visualizations of membranes, but the definitive work actually came from electron microscopy. And this is actually a couple of figures taken from Stein's book. And what it shows you is this is the outside of a cell, and this is the inside on an electron micrograph. And you can see here this clearly defined barrier, or this boundary between the two. And if you blow this region up, you see this arrangement. And this is what was referred to for quite a while in the literature as the railroad appearance. It was known, right, when they prepared, and it actually took a long time to get pictures like this. And the reason was because it took about 10 years before people figured out the proper ways to prepare tissues to reduce distortion of the tissue while at the same time being able to make electron microscope images, right? What that requires you to shoot electrons, diffract electrons off of the substance. Of course, electrons carry a lot of energy, and so they basically just burned all the tissue, and they had to figure out a way to prevent that tissue destruction from occurring with high velocity electrons actually running into it. So this requires chemical modification and the introduction of electron-dense bodies. And the fact is that the electron-dense material that's actually used to stain preparations and tissues for electron microscopy bind to regions that are known to be in the hydrophilic or be in the water phase. And so the suggestion here from this is the fact that something is hydrophilic, and there's two lines of it, and in between is a light region which is relatively non-hydrophilic and therefore hydrophobic. The actual thickness of this entire thing is about 65 to 75 angstroms, and that inner translucent region is 35 to 40 angstroms. And this turns out to be about the right dimensions for phospholipid as proposed by Overton. And so his ideas turned out to actually be correct, as we'll see in a moment. And the interesting thing over the years, once this was established, people went around and looked at every tissue, every kind of cell in every different species, and and including microbes and viruses and everything else. And the phenomenal thing was that they almost always had exactly the same structure. And that correlates actually with if you reproduce or redo Frick's experiments in terms of capacitance. Turns out that the specific capacitance, the ability of membranes in any system to actually hold charge, is virtually identical, right? So it's clear that membranes in all these different species are actually comprised of very, very similar material. And this turned out also to be true of the internal membranes. So not only were, the cell membrane, this railroad appearance, the internal membranes had precisely the same structure. And again, that was in viruses and bacteria, et cetera, and all living systems in which it was examined. So upon closer examination, it became apparent over time that these two hydrophilic bands were actually associated with phospholipids as originally proposed by Overton. And phospholipids are these amphiphilic molecules. And they, amphiphilic means loving, is philic, and amphi means both. And so it loves both the hydrophilic and the hydrophobic entities. And an example of what a phospholipid looks like here, we'll talk a little bit more about it in just a moment, but it's comprised of these tails, which are actually fatty acids. This linker region, which turns out to be glycerol, right? And glycerol is like 3 carbons with OH, 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 right? And these can actually form ester bonds. And then attached to the glycerol, the fatty acids, and these hydrophilic heads. These are referred to as heads. And they can be a variety of different things. They can be phosphates. They can be a number of different sugars, related sugars, etc. So there's a number of hydrophilic heads that can actually appear. So what you have here is this arrangement of 2 of these end to end, right? So this is the bilayer, the entire thing, and you can see here's the monolayer here and another monolayer here, and they come tail to tail, with the heads actually exposed to the water, right? And this turns out to be a very, very energetically stable, and that's the argument that Overton actually used. And ultimately, it was really this physical argument, as we're going to talk about in a moment, that led to the wide acceptance of the bilayer as the model for the membrane. Obviously combined with the fact that when you did solvent isolations, you would actually pull out lipids from membranes, phospholipids from membranes. Okay, so and then I should make you aware of this technique, which really established even further the concept that membranes are truly bilayers. So you can do these experiments where you take a cell and you rapidly freeze it, and then you can actually it's like splitting, maybe not splitting the atom, but you know how you can fracture things when they're actually frozen. If you actually come in very strategically and appropriately with a very, very sharp knife, and then pull up on one end of the broken part, what you'll actually see very often is that the um, inner portion of this ice, which you can then prepare for electron microscopy, actually pulls off. And that actually will pull off the outer fraction, the outer portion of the one monolayer, one of the monolayers. And then you can stain that, and you can also stain what remains. And this is an example of after you've taken the top off of one of these cells in a freeze fracture experiment. And this is essentially the inner monolayer, right? And underneath that is the cell. And what's been pulled off here is the outer leaflet of the lipid bilayer. And I'm going to show you another diagram of this in a moment to illustrate the differences between the inner and outer layers that actually occur. But this really shows very, very clearly that there's a bilayer system here, and it's completely as predicted because this will be extremely stable, right? And now you can actually physically pull these things apart. And so again, that's was a big part of the final, recognition and acceptance of the fact that, membranes are actually lipid bilayers. You can do energetic calculations, and you can, I think intuitively, right, if you've got these fatty acids and you try to put water here, there's going to be, these lipid faces are going to try to eliminate the water and bring those bilayers together. That strategy, by the way, and these energetic features are used, as we're going to see in a future lecture, for the production of micelles that are used for drug delivery, including actually, we're going to mention how some of the current methods for making vaccines against COVID are actually using, lipid-based methods of transfection or introduction of genetic material into systems for the production of antibodies. Okay, we'll come back to that a little bit later. Okay, so what they, I'm just going to stop. Yeah, so I forgot to mention, when you do the freeze fracture, you'll notice if you look very carefully, there's all these different nodules. Okay, and these nodules turn out to be proteins. Okay, and so the work here was consistent with the mosaic model, showing that the membrane is actually comprised of both lipids as well as these nodular protein structures on EM. And if you actually look at the composition of protein, it's highly variable actually. And the proportion of lipid to protein can be anywhere from 10 to 1 to 1 to 3. So in other words, now this is on a weight to weight basis. But you can see the wide range, and I'm going to show you some images later of a retinal cell, a rod cell of the retina of a mouse. That just gives you an idea of how much protein is actually present in some different membranes, and I have a list later of some proportions of different intracellular membranes as well. So the bottom line is that the lipid portion of membranes, the lipid to protein proportion, is extremely variable. And moving forward, we're going to first talk about the lipid composition and bring out some features of what's important about those. And then we'll come back and talk a little bit more about the protein end of membranes. So one of the main compositions on the lipid side of membranes are, as I've already said, phospholipids, discovered by Overton originally. I mentioned already that these are amphiphilic and they have a head to tail appearance. And here you can actually see much more clearly the glycerol backbone, which is actually shown here. And you can see that there's an ester link. There's two available for linking up a fatty acid to that glycerol. And then the third location actually can link up typically a phosphate to which is actually attached a variety of different moieties and entities. And that's why they're called phospholipids, right? Because they all have this phosphate group that's actually attached to the glycerol. And here are some examples of the main actually phospholipids. So first of all, phosphatidic acid, that's just a phosphate on the end, very important um, component of almost all membrane systems. You've got phosphatidyl um, ethanolamine, phosphatidylcholine, phosphatidylserine, phosphatidylinositol. And there's some others, but these are the primary ones. And you might have heard of some of these before, right? Like a lot of nutraceuticals include phosphatidylserine and phosphatidylcholine, right? People take these for better memory and all kinds of stuff. There's all kinds of hoaxes out there that suggest that if you eat these things, you become smarter, wiser, I don't know, stronger, all kinds of things. But in that particular case, you're really eating something that supposedly is ultimately getting incorporated into the cells of your membrane and actually having an impact on cellular function. Now, these different phospholipids can have a variety of different fatty acids attached to them. So here's again the structure of a phospholipid with the two fatty acids. And we refer to those fatty acids as, this is actually not correct, has 16. C2 refers to this one, C2, and this one here refers to C3. Okay, so make a note of that from the notes, because I think it's actually not clear in the notes as well. Now, it's true, though, that the C3 does tend to have 18 carbon fatty acids and C2 16, but that's not an absolute rule. Okay, so C2 refers again to the position 2 on the glycerol forming the ester bond, whereas C3, right? So and the C1 actually binds to the phosphate in the case of phospholipids. Now, the fatty acid components themselves can be referred to as saturated or unsaturated. I think you probably have heard of this before and know about it, but unsaturated just means that this long chain, I didn't mention this before, this is a long hydrocarbon chain. So each one of these vertex is actually a separate carbon, right? And they're completely reduced. So they're just hydrogens bond, two hydrogens, carbon, and then two hydrogens coming off. In this particular case, of course, you're missing, two of the hydrogens, right? And that's what we refer to as an unsaturated bond in the fatty acid itself. So that's how we refer to saturated and unsaturated fatty acids. And you may remember this from your organic chemistry, but when you have an unsaturated bond, it's actually a double bond between two carbons, because for quantum mechanical reasons, they actually form a plane, okay? And the carbons can't rotate around each other, at least not easily. And so the net result of this is that this actually creates a kink in the fatty acid side chain. And that's actually illustrated here. You can see that this comes down and then makes a sharp bend. Now this thing can rotate around in this fashion, right? But that kink tends to remain because of the planar relationship of these two carbons that actually exist. Whereas in an unsaturated or a saturated system without double bonds, all these bonds between different carbons can rotate easily. And the net result of that is that a fatty acid tends to remain in a linear, for energetic reasons, in an actual linear plane or a linear arrangement. Okay, so these are quite important to appreciate, as we're going to see in a moment, because this has a lot to do with fluidity of membranes, right? Membranes are in the plane are actually a little bit like a liquid. And it's the amount of unsaturated fatty acids that determines that. And the other thing I'll just mention here is this idea of polyunsaturated fatty acids. So many of the fatty acids don't just have one kink, they have multiple kinks, right? And we refer to that as polyunsaturated fatty acids, or PUFAs. Okay, and then here I just have listed some, various combinations of different arrangements. And you can see oftentimes C2 is 16, but you can see it becomes 18, and it can really be anything, but these tend to be the most common, them being actually 16 on C2 and 18 on C3. Okay. Yeah, and just a little mention about PUFAs. You've heard of these, I'm sure. There's a lot of, again, nutraceutical nonsense about omega-3s and omega-6s and how important the omega fats are for a person's health. I don't know how much of that is true. I can tell you that when people have studied the impact of people taking these supplements on in the cardiovascular system, I believe there isn't a single credible study that has actually demonstrated any net benefit. So my suggestion is just save your money, but you may know of other studies, maybe in the neurosciences or other areas, where they have shown actual true benefit of the omega fats. Bottom line is that, if you want to get these, you can buy them as a powder or a liquid, I should say. But the easiest way to get them is from fish oils. It's much, much cheaper. And it's actually, you need fish oils from the fish in the northern Atlantic in particular, but in cold waters. And the reason for that, well, first of all, here are the omega. First of all, I'll just give you a definition of what an omega-3 fat is. An omega-3 fat means that there's a double bond at the third carbon position. Okay, from the end. So they're labeled from the end, 1, 2, 3, blah, 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 right? And then there'd be, like, if this was a C16, there'd be 16 of these carbons. And between 3 and 4, there's a double bond, right? That's a very common place to actually see a double bond on one of the fatty acids. And if there is one on position 3, it's referred to as an omega-3 fatty acid. And they're very often PUFAs, right? They often have multiples, but they don't. The number of these are highly variable. And then the omega-6s are actually another class, and they have the very first double bond at position between 6 and 7. Okay, so that's how they're actually defined. And I mentioned here again that they're of no limited value, if any value at all, in cardiovascular patients. Okay, besides phospholipids, there are other kinds of lipids that actually exist within membranes. And they're listed here. So I've covered here already the phospholipids, and there's the sphingolipids, the ceramides, the sphingomyelins, cholesterol, and there are a host of others. I'm just showing you here the different, like sphingosine, for example, the sphingolipids, ceramide, sphingomyelin, cerebroside, and there's a whole host of these other ones that I haven't actually listed. I'm going to come back and tell you a little bit more about these because they're very important actually in signaling. And then the other component that's very important to think about is cholesterol. Okay, cholesterol is an important component of membranes, and as we'll see in a moment, it, along with the unsaturated fatty acids, have a huge impact on the fluidity of the membrane, and that actually impacts on the function of the membrane. Okay, and this is just really, this I wouldn't expect you to know, like don't freak out. This is just to make a very important point. You've all taken cell biology and you've all taken about cell signaling, right? And signals come from the outside and they go into the inside, and where does that transduction cascade begins at the membrane. And the membranes have lots of these receptor proteins that are actually residing. And I've already told you that about, between 20 and 30% of the genome is actually dedicated to membrane proteins. And many of those are actually receptors, right, on membranes. And there's really two large classes of these receptors. And these are not, there's others too, G-coupled protein receptors and receptor tyrosine kinases that reside on cells. Now, they're very, very important, obviously, for obvious reasons. And you've taken all about that. But, you might know less about these more primitive signaling systems. So if you look in the very primitive organisms, much of the signaling actually originates from lipid metabolism. And this kind of makes sense, right? That you see this in bacteria. You see it in all the different life forms. Will have a fairly important or signaling will through lipid metabolism will be a very important aspect of regulation of growth and responses in those organisms. And I just mentioned here that targeting things in lipid metabolism is big business. And things like Advil, aspirin, the COX inhibitors, those are the ones that come to mind immediately, are all involved in manipulating lipid metabolism. And just to give you some examples here, what I'm showing you is the ceramide and sphingosine metabolism and how it actually gives rise to this thing called S1P, which is a very, very primitive but important signaling pathway. And here again are the ceramides just linking up again to the sphingomyelins, producing a variety of different compounds that are involved in apoptosis and ROS and inflammation. Cholesterol itself is subject to lots and lots of metabolism. Many of the steroidal compounds are derived ultimately from cholesterol metabolism, leading to testosterone and estrogens and all the sex hormones and a variety of other kinds of hormones as well. Here's an example of the phosphatidylinositol molecule. And it's in a very, very complex array of enzymes that produce all these different factors, many of which will impact on very, very specific downstream elements and proteins. I actually spent a fair bit of my life looking at PIP2 and PIP3, these two, and looking at their regulation in the heart. And you can see here that they have an impact on PI3 kinase, is something that converts PIP2 to PIP3, and this is very much involved in activating pKa or AKT, which is a protective enzyme that phosphorylates a variety of different targets and has a big impact on a variety of different kinds of remodeling. But again, the point of this diagram is to emphasize that the lipids are not just there for creating this barrier. They're actually very much engaged in signaling. And when they get metabolized, you get a variety of different responses as a result of this metabolism. Okay, and now I'm coming back a little bit to talk about this idea of fluidity and movement of lipids within membranes. And I already mentioned that fluidity has a lot to do with unsaturated fatty acids as well as cholesterol. And a point I want to emphasize here is that fluidity is an important factor in determining the function of the proteins that are actually in the membrane. They also have a big impact on the permeability of the membrane to a variety of different substances, and we're going to talk about that in the next couple of lectures. We're going to do a sort of profiling of the things that can make it across membranes and why, and we're going to discuss sort of why certain things get across and why other things don't, and the factors that contribute to that, temperature being an obvious example of that. Now, in the case of homeotherms like ourselves, warm-blooded creatures, our lipid composition really doesn't change very much from time to time and from cell to cell. It probably does during development, but things are relatively static. Where this becomes of extreme importance is in poikilotherms, where they don't control their temperature, cold-blooded species. And if you every summer winter cycle, there's a huge change in the composition of membranes for a number of species, particularly fish, frogs, and other cold-blooded species. And the general law is that, or the general feature is that they get more and more polyunsaturated fatty acids in their bilayers as the temperatures drop. And again, the idea of this is that these kinks actually allow or prevent the lipid bilayers from packing. And this is a less fluid or more solid-like state than is this, right? This is more fluid-like because these things can't really pack together. And we think of solids as being things that are more crystalline in nature. And so increasing the number of polyunsaturated fatty acids creates more of a situation like this, which ensures that proteins in these membranes can continue to function. There are lots of studies that have been done in terms of looking at, the Q10 of various enzymes, membrane enzymes, and it's very clear that when they tend to shut off, it tends to happen over a very small temperature range, which actually coincides with essentially a phase transition from an unordered state of the lipids to a much more ordered state within those lipids. And then this diagram is just shown in here again to illustrate the idea or remind you that cholesterol also has a big impact on the relative fluidity of these membranes. Yeah, so now we're going to switch over and talk more about the protein components. And I've already shown you this freeze fracture technique, which clearly illustrates the mosaic nature of membranes. And I showed you this before. This is the inner membrane monolayer, and you can see all these nodules. Now, if you actually look and compare now a blow up, this is not necessarily the same one, but of the inner cytoplasmic layer and the outer monolayer, you can see a huge difference, right? And this is actually telling you that there's much more protein associated with the inner membrane than there is with the outer membrane, right? Has considerably less protein. And much of this protein, by the way, is, as we'll see in a moment, not integral protein. It's actually just sitting on the surface, okay? But this is a general fact, right? And of course, that sort of makes sense because inside the cell, you have much, much, much more protein than you do on the outside. And that's actually reflected on the proteins that you actually can, or the number of nodules that you can actually pick up on the monolayer portion of the lipid bilayer. Here's, I mentioned already this before, if you actually look at the proportion of lipid to protein in membranes, what you find is, of course, there's many, many more lipid molecules than there are proteins. And that's because, though, the molecular weight of lipids is much less than the molecular weight of proteins. If you go by on a weight-to-weight basis, I threw these numbers out here before, you can have a ratio of lipid to protein that goes from 10 to 1 to 1 to 3. So, virtually 3/4 of, in this case, is actually protein. Okay. And so in terms of surface to volume, you'll find that the numbers that you'll read are 10 to 60% of the membrane can actually be occupied by protein. And here's an example of some examples of the proportion by weight of protein to lipid in different kinds of cells and in different membrane preparations. So you can see here the red blood cell is approximately 2/3 protein, right, relative to lipid. Liver cells are about 50-50. Amoebas are, the opposite, right, about 1/4, 3/4. And then the one that's really striking is the inner membrane of the mitochondria. And you can see it's 3/4 more than 3/4 of it is actually protein. Similarly, chloroplasts, right? And of course, these are the centers for energy production here and energy uh, utilization here in the mitochondria. Okay, and then the other thing that's actually in Stein's book that I thought was quite interesting was the idea of the use of the atomic force measurements. And these are things, are these very, very sensitive cantilevers. So what you do is you prepare tissue, you fix it typically, and then you, with a piezo system, actually, you can move the structure very, very slowly across a little lever that moves up and down and just rides the surface of your preparation. And this is actually the example I told you I would show you of a membrane of a rod cell in the retina of a mouse. And you can see how it's just this very orderly arrangement of these nodules. And these nodules turn out to be the protein rhodopsin. And rhodopsin is actually a G-coupled protein receptor that has in it a vitamin A derivative that actually absorbs light. And when it becomes excited, it leads to the activation through this G-coupled protein receptor, through the G proteins, actually activates a phosphodiesterase, which reduces cyclic AMP or cyclic GMP, which then closes a channel that causes that rod to actually reduce the amount of neurotransmitter release, and it actually leads to the so-called that's the detection mode for light in that system. Okay, so a rather and so the point is that a system like that has an enormous amount of protein, right? Because you've got a cell that's just detecting as much light as it possibly can. And so the membrane is essentially chock full of these receptor light receptor proteins within the membrane. Okay, so I just took a little break to have some water there. Okay, so now I just want to mention that the proteins that are actually associated with membranes can be either what we refer to as embedded proteins, or they're associated or peripheral proteins. Embedded proteins are integral proteins, are also referred to as integral proteins. And integral proteins essentially are, as shown here, they actually go into and are embedded right in the membrane. That's why they're also called embedded. And I want to make a point, I'm going to make this point later in the course several times, that almost all these proteins, they're shown here as if they're in isolation, but invariably, most of the important proteins that I'm aware of are in large macromolecular complexes. So you should be thinking about this as, there's not just one protein here. There's like 10 or tens of proteins that are self-assembled. And we'll come back and talk a little bit more about that a little bit later in the course. Membrane proteins, certainly the integral proteins, are invariably glycosylated. You can look that up from your cell biology. That just means that they have stuck at the lysine residues or at the serine residues. Typically, they've actually got a chemical covalent bond between those side chains and carbohydrates, various types of carbohydrates. And these turn out to be very important, as I'll mention a little bit later, in cell recognition. The key is that the integral proteins have essentially a hydrophobic core. So they have some kind of a structure that allows them to be very stably inserted into the membrane. And you're going to see how that plays out in just a moment. The integral proteins, or the peripheral proteins, as well as the integral proteins, actually, this is not really all that accurate. It says that peripheral proteins often attach to cytoskeleton, but that's also true of many integral proteins, although the peripheral proteins, this is more so true, if I can use that term. And as we'll see in a little while, this connection of membrane proteins to the cytoskeleton gives the membrane a lot of strength, which it would otherwise not have. A lipid bilayer, think of it, you can think of it a little bit as a soap bubble. You know, you've probably all done this before, where you take soap, put it into water, and then create a, you know, a ring structure, a circular structure that you then put the, and then you then put this into the water soap mixture, and you can blow these bubbles. And you know, when the bubbles are large enough, they actually are relatively unstable. And that's consistent with a lipid bilayer, actually, those, the appearance of those bubbles. And they're actually quite weak, right? You can break them quite easily. But real membranes have got these proteins inserted that are physically connected to struts, right? To cytoskeletal structures, which give the membrane form and strength, and prevent them from getting blown up every time there's an osmotic stress that actually occurs. These proteins, by the way, are also amphiphilic, right? Because you're going to have, typically, there's going to be a core region here that's actually hydrophobic, and then the ends of these are going to tend to be hydrophilic, right? Because they're at the water interface, so therefore it would be logical energetically if they had lots of polar side chains. And that turns out, in fact, to be the case. It's one of the ways, as we're going to see in a moment, you can distinguish between, by just looking at the primary structure of a protein, what regions are likely to be in the membrane and which regions are not. By the way, there's a duplicate slide that was left over. Please remove the first version of this slide or this page in your notes, and we'll just focus on the second one. So this is just coming back to the point that, you know, if you look at an integral membrane protein, you find that there's a region that actually inserts into the membrane. And these regions have got, almost always, but not 100% of the time, have got an alpha helical structure, as shown, as illustrated here. And in this particular case, we actually have two proteins. This is very common, where the N terminus is on the outside. They are single transmembrane proteins in both cases, right? We're going to refer to these sections of the protein as the transmembrane region or segment. It's more common to be called a transmembrane segment. And you can see there's two of them. Each one of these has one. And in each case, there's a C terminus on the inside. And many membrane proteins are like this. They span one time, and they've actually got the N terminus on the outside. And that has to do with the way proteins are actually produced at the Golgi. But the thing I want to focus in on is this region, this transmembrane region. And you can actually, you find that there's an alpha helical arrangement of the amino acid backbone to these transmembrane regions. And that's actually illustrated here, where you can see a peptide bond here. Okay, and you can, if you look carefully here, if you kind of count, turns out that to form this, a very stable structure, chemical structure in alpha helix. The typical alpha helix is going to have a pitch, what's called a pitch, which means that there's essentially 3.6 amino acids in one pitch. So a pitch would be from, you know, a 360-degree turn of this alpha helix is a pitch, right? And there's, from this region to here, on average, there's 3.16 amino acids. And this is the peptide backbone. And emanating from that are these R groups, right, or the side chains. And in these transmembrane regions that form these alpha helices, the side chains are invariably hydrophobic side chains. So these R's will be highly hydrophobic, right? Very well-defined structure. Out here, you can have a variety in the water phase. You can have a variety of different structures. There could be hydrophobic structures, for sure, hydrophobic side chains, which, by the way, will tend to, you know, find one another and create a structure based on hiding the hydrophobics from the water. But here's where you're going to find a much greater probability of finding a hydrophilic side chain, right? And hydrophobic side chains would be like tryptophan, isoleucine, leucine, alanine. Those are relatively hydrophobic. The hydrophilic ones are obviously lysine, aspartic acid, glutamic acid, right? The charged side chains. You should know this from your biochemistry, I would think, I would hope. Okay, so the nice thing about recognizing, that there are these integral proteins is that you can actually predict from the primary structure of a protein uh, what the regions are, the likely regions that actually insert into the membrane. And some proteins insert multiple times into the membrane, right? They go back and forth in and out of the membrane. So they have multiple transmembrane segments. This idea was formalized by Kyle and Doolittle. I think it was in the early 60s. I don't remember the exact time, but they came up with this idea of doing hydrophobicity plots, hydropathy plots. And then again, essentially the idea is I've got a protein. I know the gene structure. So from the gene structure, I can determine, right, the sequence of amino acids, right? Amino acid 1, amino acid 2, amino acid 3. And the idea of a hydropathy plot is that we just score every one of the amino acids, of which there are, I believe, 20, yeah, the 20, no, 20, 23, 20 amino acids, yeah. And by the way, I forgot to mention on the previous slide that, whoops, I went the wrong way. On the previous slide, I forgot to mention that in this alpha helical structure, there are about 20 amino acids, okay, in a transmembrane segment. Okay, so what the hydropathy plot does is it actually looks at sequential amino acids, and you just move along the length of the protein. And for the 20 amino acids, you just add up the hydropathy numbers. So if the amino acids are, for example, methionine, if this is a methionine, and let's say a proline, you would just add the hydropathy numbers. You do that for 20 amino acids, which is about the number that are required for a transmembrane segment. And then you plot that. You plot that hydrophobicity as a function of position along the protein. And what you can see in this particular case is there are these regions that have very high hydropathy scores. And the prediction would be that those are the regions that actually insert into the membrane. This happens to be a hydropathy plot. This is provided in Stein's book for lactose permease, which is a E. coli or a bacterial transporter that picks up and transports galactose into the bacteria. And in this particular case, it corresponds to the number of large hydropathies here. I think it was something like 12. Which corresponds to the actual number of transmembrane segments that are actually found in the lactose permease. So we're going to come back and talk about that when we introduce some of these different channels and these different transporters, because the early work actually predicted already some aspects of structure, which turned out to be of critical importance for the understanding and the ultimate structure of these particular proteins. So, but very straightforward, right? It's an integral protein. That that's in the membrane. The energetically most stable arrangement of that is an alpha helical arrangement of the side chains, exposing the side chains to the lipophilic region of the lipid bilayer. And of course, those would be hydrophobic residues that will tend to insert into the hydrophobic region of the membrane. Okay, so that's all sort of talking about static structure. And I just want to mention a little bit now about dynamics of membranes. Membranes are not static. Of course, we're living in at finite temperatures. and there's a lot of thermal energy which is contributing to motion. And membranes are therefore, like all things, are highly dynamic. And if you actually look at the dynamic properties of these lipids, and we're talking now here about lipids, we're focusing on lipids, they're able to move laterally. So any one of these phospholipids or the ceramide or these other molecules that are actually inserted into the lipid bilayer, they're able to move laterally. It's like a fluid there. And they're able, therefore, to diffuse. And the typical measurements have been shown that they move about 150 to 100 nanometers over a millisecond, which is a substantial speed, actually, relative to even things that are in the aqueous phase. And I've already mentioned the idea that the unsaturated fatty acids make things more fluid. And sure enough, if you play around with membranes and you actually introduce more unsaturated fatty acids, you will increase dramatically the lateral diffusion of these lipid molecules within the bilayer. These molecules also are able to rotate, and so they're spinning and moving their fatty acid side chains in circles at the same time. Those are even faster than the lateral diffusions. And these involve vibrations as well as bending motions of the fatty acid. So all the different possible motions that are available to the tails will take place. And the amount to which the tails actually move really dictates how rapidly and whether the molecules in the membrane will rotate and the extent to which they will actually diffuse laterally. The importance of this turns out to be primarily that when it's moving rapidly, it actually contributes or modifies and modulates the activity of proteins. And we'll come back and talk about that. The bottom line is that movement increases as temperature increases, and we sort of know that, right, intuitively, that that's also the case for systems that are other than membrane systems. And here's the point I wanted to make in terms of the relationship between protein activity and fluidity of the membrane. So there in the 70s and the 80s, a lot of NMR spectroscopy, nuclear magnetic resonance spectroscopy, was done to measure how disordered lipid bilayers actually are and to correlate that actually with protein activity. And the observation is very clear that there are these critical temperatures at which you really undergo essentially a phase transition. So this would be very much like ice and water, right? And we know that, you hit 0 degrees in pure water, at least, and you very quickly, over a very short range of temperatures, you're going to go from a water phase, a liquid phase, to a solid phase. And the same thing actually happens within membranes. And in this case, when we talk about order and disorder, right, fluid versus non-fluid, we're really talking here about the lateral diffusion of these phospholipids and lipid molecules within the bilayer, as well as the amount of motion of the tails, right? That's what dictates, it going into the solid phase. The critical thing here is that when these membranes were shown to go into this ordered phase, the amount, if you were to plot here at the same time, the actual activity of a protein, it essentially follows the same sort of path. So telling us that the activity of proteins and enzymes are highly critical, are highly dependent on the actual physical state of the lipid within the lipid bilayer. But none of this happens, throughout the membrane. Some regions will actually freeze and other regions will not. And you should think of membranes very much as ice, they're like icebergs. They have like icebergs in the ocean. There's regions that are relatively structured, and then there's lots of regions, most regions that are very unstructured. If you take the temperature down far enough, you can force most of the membrane to actually go into a very structured arrangement. Okay, so the last thing I just want to call to your attention is that most of what we've been talking about, well, most of the things we're going to be talking about in this class will be related to, fluidity of membranes with respects to lateral movement and that sort of thing. There is another kind of movement that actually takes place in membranes, which is much more controlled, and that's called the flipping motion. And that is the ability of a lipid to actually change its orientation. So if this is the outside, you can actually get these lipids to flip from being outer faced, where the head is on the outer side, to where the head is actually on the inner side. And this is highly controlled by a series of enzymes that are called flipases. I guess that shouldn't surprise you. And it's important because if you actually take a look at the orientation of membranes, they're not random. Most of the ethanolamides and the phosphatidylserines are actually sitting on the inside, whereas the phosphatidylcholine and sphingomyelins are actually facing the outside. And when phosphatidylserine actually flips to the outside, it's actually a signal to the outside world that this cell is in distress. It happens to drive inflammatory processes, and the net result is that the health of the cell is very much monitored by, the orientation of the phospholipids within the membrane itself. The cell actually spends a fair bit of energy making sure that the arrangement is phosphatidylcholine and sphingomyelin on the outside, and ethanolamine and phosphatidylserine actually on the inside. Okay, the last thing I'm going to mention with respects to sort of movement is the movement of proteins. And they also move. And a common technique that's been developed over relatively recent years is the technique of fluorescence recovery from photobleaching, or FRAP. And so what you do in this particular case is you put a fluorescent label onto a protein. And the most common protein that's actually used is green fluorescent protein. It's a very common strategy now. You can actually, get zebrafish, you can get mice, you can get, rats, you can get other species where a variety of different proteins have actually been labeled with fluorescent labels. You've got, mice running around with green brains and green hearts and green blood vessels for labeling purposes. Now, this technique has been used in membrane proteins, and it allows for this following kind of experiment. What you do is you have a protein with a label, you're interested in its movement in the membrane. You hit it with a very, very strong amount of light, which causes photobleaching, which basically destroys the fluorescent label. And then what you do is you just wait and see how long it takes for that region that you photobleached to actually fill back in with fluorescent material, which would be coming from other regions of the membrane. And so, an example is shown here where you look at the intensity of the fluorescent light that's coming from that cell in this particular case, or that portion of the membrane. And you can see that it drops with photobleaching, give a strong bit of light, and then you watch the recovery to actually occur. When those experiments are done, what is observed is that there's a massive range of mobilities, ranging from proteins that move almost as quickly as lipids to proteins that take several minutes to actually, like this time constant could be several minutes or even longer. And an example of that is actually this BAN3 protein, which has been looked at. And the reason that this protein moves extremely slowly is because it's an example of a protein that's actually bound to the cytoskeleton. So it's kind of anchored into whatever region of the membrane it gets inserted into. And from there, it has a very, very hard time freeing itself to be able to move between different regions of the actual membrane. All right, so that sort of deals with the concepts of dynamics of membranes as well as the structure of membranes. I just want to sort of end the lecture talking a little bit about the idea of heterogeneity. So we know it's a mosaic, we know it's heterogeneous from the standpoint of there being many different substances, but that doesn't necessarily mean that, that there's notable differences, regional differences from region to region on a membrane. For example, if you look at that rhodopsin figure that I showed you before, there's a very clear pattern that actually exists. And so it looks relatively homogeneous. It's not homogeneous, but the structure is sort of predictable. Um, there's actually, though, lots of heterogeneity, both in terms of function as well as structure within membranes. And that results in membranes being much more permeable to certain substances in some regions versus others. And also means that certain kinds of functional activity is very different from one region of the membrane to the other. In later lectures, we're going to talk about polarized cells, where there's a completely different kind of activity taking place on one side of the cell versus the other. And things like aquaporins are very, very critical for that kind of thing. Like cells move essentially by hydrodynamic flow, and they use aquaporins in order to be able to do that. And we'll talk about those kinds of things in later lectures. And so that's already that's a preface for the concept that there's a lot of regional heterogeneity that actually exists within cells. And I'm just going to give you a couple of examples, the most well-known examples of that. And those are the invaginations that are associated with calveoli and the clethorin-coated pits. And calveoli are areas that are quite important in a lot of signaling, especially G-coupled protein receptors. If you look in, for example, muscle cells like cardiac cells, and you look deep in the T-tubules, I don't know if you know what those are, but they're invaginations of the lipid bilayer deep into the cell. And they are sort of full of caveoli. And those are areas of high density G-coupled protein receptors. And so that's one of the purposes, it appears, of the caveoli. But in the case of muscle cells, they're probably of equal importance is the idea that they act as kind of a reservoir of membrane. And the way you can think about this is illustrated here, where you might have a cell that is in a resting state that needs to undergo an elongation. And when this occurs, of course, there's a big change in the surface to volume ratio, right? Which means that and cells are not changing their volume very quickly, right? So that's kind of a fixed entity. You have to move things in and out across the membrane in order to change the volume. When things undergo a contraction or an elongation or some change in form and shape, you're going to have that same volume now, spread out over a much different surface area. And just to give you an example, right, if you were to minimize the surface of an object, a three-dimensional object, turns out that that's a sphere. That shouldn't surprise you too much. That the sphere maximizes the volume for the amount of surface that you actually have. Now, if you turn that into, a cylinder, you now have, for the same volume, you have a much large increase in the actual surface area. So cells are constantly changing their form. Like every time your heart contracts, every cell is actually changing, its surface to volume ratio by up to 30%. Skeletal muscle, even larger changes actually occur with stretch and with contraction of the cells. So one of the ways to deal with the rapid changes in surface to volume ratios is to have these reservoirs of membrane. And that's one of the purposes of caveoli, is that they actually provide that extra surface that's actually required. So as the cell swells, let's say with osmotic swelling, for example, you can swell cells very often, most cells, by at least a factor of 2 without actually bursting those cells. And they have reservoirs of this extra membrane. And on the right here is actually showing you what this actually looks like. Um, and so in the T tubules in particular, in cardiomyocytes, I mentioned already that they're chock full of caveoli, and that's where the extra membrane actually sort of is believed to come from, or largely comes from, when there's a change in the shape of a contracting muscle cell. The other, and so these are very heterogeneous, right? You don't see them, you only see them primarily in the T tubules. It's very clear that this region of the membrane is very different from other regions, right? That's what I mean by heterogeneity. And caveoli tend to also be areas of higher permeability to certain kinds of substances, including ions. The other one I just want to draw your attention to are the clethra-coated pits. I don't get into too much detail on these, but these are highly dynamic structures, and an example is shown here of a clethral-coated pit actually forming and then invaginating, and then eventually separating from the lipid bilayer. And you'll notice here, this is a membrane, right? This is still a phospholipid, and now what you've done is this material that has been either in the membrane or sitting on the surface of the membrane is now encapsulated in here. This is a very, very common method for actually turning over membrane proteins. So, there's a process called desensitization that happens. If you drive receptors too strongly, they become phosphorylated internally, and that often leads to a down regulation, right? It's like a negative feedback system that often leads to internalization of those receptors into these vesicles. And very often these vesicles will actually recycle back to the surface when the signal is gone or dissipated somewhat, or it can send this off for degradation. Okay, and it's a way to actually do quality control of proteins as well. And that often involves these endocytic invaginations, these endocytic endosomes fusing with lysosomes, which then lead to the destruction of the material that's actually in the endosome coming in from the coated pit. And I should also mention that these are the areas that are involved in cholesterol uptake as well, right? From the lipoproteins, many of these regions that are clethrin coated are the regions where the receptors for these lipoproteins actually exist within cells like heart cells and liver cells. And this is how these things actually make their way into the cell. Same thing with ferritin and iron metabolism, and a number of other processes for transport actually involve clethrin-coated pits. So this is our first example of transport in membranes, right? We're talking now about big things going through. Some of it's recirculating and quality control, but in the case of iron and cholesterol, they are actually getting in and out of the cell through a very complex process that involves many proteins and lots of energy. We're not going to focus on these kinds of transport processes, although we will mention them in an upcoming lecture in a day or two, in a lecture or two. I'm almost done. Okay, and I just want to sort of end on this slide. There's a couple others, but the main, where there's some information transfer here. Membrane proteins, like we classified the different lipids into different kinds of lipids. Proteins are also classified in terms of different kinds of proteins, membrane proteins. And they are transporters, which include channels and carriers, and also and we are going to, and we can include pumps in here as well. And this is what we're going to focus on, right? But there's other kinds of membrane proteins as well. There's things like, receptors. We already talked about those G-coupled protein receptors, receptor tyrosine kinases. It's a whole host of others. So lots of enzymes, right, that catalyze a variety of different reactions. In fact, the receptor tyrosine kinases are enzymes themselves. And in some ways, G-coupled proteins are also enzymes, right? Because they catalyze the activation of G proteins. Um, but nevertheless, they are enzymes. They're a separate class. There's also the cell adhesion molecules that are involved in sort of holding cells together. This becomes particularly important in skin, but also just forming tissues, right? Being able to keep certain groups of cells together to even organize within tissue, right? Blood vessels from parenchyma of the different tissues. You require these cell adhesion, and there's a whole slew of these, right? And it's like a lock and key. Provided the neighboring cell has the correct lock, the key will fit in from another cell, and this is how you get cell recognition and the creation of tissues. And then there's these docking proteins. These are like the anchors, right? And I've already talked about how the membrane itself is strengthened by its association with the cytoskeleton. And the way you actually link up and really make that a strong connection is through these anchor proteins. And the way these anchor proteins actually work is they've got a inside and an outside, and they connect to sort of extracellular matrix out here, and they connect to the cytoskeleton in here. And so now you've actually got a cementing across the cell membrane of the cell structure with the outer structure of the structure in the outer regions of the cell. And then the last group are these self-identity markers, right? Which are used for the HLAs and for immunity purposes, for surveillance of cells, etc. And they're very often link up with foreign proteins to initiate reactions and immune-based reactions for the purposes of surveillance of foreign materials. And I guess I have some examples here. I think I'll just bypass these and end my lecture by just saying, look, long discussion about membranes, really coming in on and homing in on this idea that the membrane is a fluid mosaic of lipid and protein that leads to a very complex, diverse a variety of different functions, and we're going to talk about those functions in terms of transport. Okay, so I don't really know how long I've been talking for, but it seems like a long time. I hope the message of this lecture is quite clear, and I'm happy to discuss it. It turns out tomorrow further. Okay, have a good day.